So I want to give you a little bit of history of how we started with Metabolomics Workbench. It's been a long 20-year journey, starting with lipid maps. And Ed Dennis, who was one of the pioneers with lipid maps, Glue Grant, is present in this audience. And so we started in 2003 with NIGMS when they funded us for Glue Grant. This lipid maps continues with Wellcome Trust Grant and now continues with another grant. However, this led NIH, especially NIDDK. I really want to reach out to give kudos to NIDDK. NIDDK really pioneered this effort. In 2000, they realized that this is extremely important for the larger national metabolomics community. So they initiated a large common fund initiative in 2012, and this led to the creation of the Metabolomics Workbench, and which continues to date. And it's really it is the main resource for metabolomics in the United States, and it is considered to be one of the top resources in the world for metabolomics community. And so this is an effort which involves a lot of people in my group over the years who have been involved. And this includes Owen Fahey, who is one of the coordinators of the Metabolomics Workbench grant, as he was before for lipid maps. And then we have a large number of people who have contributed to the continued growth of this Metabolomics Workbench. I'll now move on to tell you about what Metabolomics Workbench is about. So why metabolomics? We know the central dogma in biology, DNA gives RNA gives proteins, and so this is something well known, but what central dogma doesn't explicitly state is the involvement of metabolites, and metabolites are involved in everything from they are the functional endpoint of physiology and pathophysiology. You can measure it in things like serum, from saliva, from urine, from blood, from feces. And so this is really easily accessible body fluids can provide provide information about the state of physiology or pathology. And we know this very well. And in fact, metabolites can be, you can think about taking medication or drinking beer or whatever it is, which gives you really changes, alters the metabolism in the humans, and these can be measured. It's direct mirror of environmental influences, for example, malnutrition, exercise, medication. In fact, NIH now has a very large grant on a very large set of grants, one for nutrition and one for exercise. The nutrition for precision health, which is part of all of us, and exercise, which is called Motor Pack, where they are looking at exercise physiology and consequence to blood, muscle, and adipose tissue in humans. And so we'll see more and more of this as time progresses, and this has really become now state-of-the-art and standard for measuring human physiology. See, if you look at the growth of number of publications, I only have this till 2022, but in fact, when I extrapolate it, it's grown even more exponentially. There are over 200,000 publications per year in metabolomics, and this continues to grow. And we created the Metabolomics Workbench, and when we started the Metabolomics Workbench, there were no other national resources. There was an international resource called Metabolites from EBI, where people had some few studies, but it was really not very popular, big resource at that point in time. So NIDDK and other institutes jointly created the Common Fund Initiative, and this gave birth to the Metabolomics Workbench. The Metabolomics Workbench is a website, metabolomicsworkbench.org. Many of you in this audience are probably familiar with it. If not, you should visit it. I call it as a biologist-friendly website because you can navigate the website with absolute ease without knowing sophisticated programming or software engineering. You can upload studies, manage studies, browse studies, search studies, analyze studies, everything in one environment. The power of something like a workbench is not just a database. It really enables a researcher to be able to interactively analyze and download the results and learn biology in the process. And so this is an example of where as of yesterday, 2/27/25, there were 3,674 studies, some studies as large as 10,000 human patient data. And these have been all processed by us in the National Metabolomics Data Repository. And of these, 12 are publicly available, and the rest will become available in progression of time. And so they'll be, as soon as the embargo is relieved by the PI who submits the data, we'll release the data. If you look at the total number of studies, I just pointed out to you over 3,600 studies. Number of human studies, incredible, 1,480 studies. Some studies have, as I mentioned to you, 5,000 to 10,000 humans. Number of samples we have processed so far in these studies is 381,334. Number of named metabolites is 801,864. Number of data points is in millions, 64 million, and so forth. And so this really, a number of raw files, we have about 500,000 raw files in the Metabolomics Workbench. The growth has been exponential, as I pointed out to you. This is July 24, and we keep increasing. And every day, on average, we get between 2 and 3 studies that are uploaded to the Metabolomics Workbench. The number of studies in humans is the largest. It's about 1,338 studies, followed by other mammalian species, mouse and rat. These form the largest number of studies in the Metabolomics Workbench, providing a direct insight into ability to look at the normal and pathophysiology in mammalian systems. Cancer dominates the study with 513. However, diabetes, the DDK is not far behind. Diabetes, obesity, fatty liver disease, these are all very close, a large number of studies, and even cancer has a lot of cancer studies associated with organs of importance to DDK. Blood happens to be the largest source with 814 studies on blood, followed by liver and cultured cells and liver. Liver has a large number of studies in the Metabolomics Workbench. So the number of metabolites, lipids constitute the largest number of metabolites in the Metabolomics Workbench studies. As you well know, lipids are a major component of human physiology, and we have 60% of human physiology has lipids as the main components. So we have followed by amino acids, which is about 20% roughly, nucleic acids, carbohydrates, other species, and alkaloids. So lipids really form a major component of the Metabolomics Workbench. I now want to walk you through developments in the Metabolomics Workbench, which will help the community, and then walk you through 2 really concrete examples of how you can take Metabolomics Workbench to research as a researcher, biomedical researcher in the community. First, we developed what is called the RefMet, and I'm going to explain to you what RefMet is. When we started Lipid Maps project, and Ed Dennis will remember this, the last time any standardized nomenclature was developed was 1968, which is IUPAC, which is really, ancient, and it really did not account for the large number of metabolites that were developed, that were present at that point in time. So we said our first task is to create a standardized nomenclature, and this is what we did with Lipid Maps, and this gave rise subsequently to all of metabolomics. We developed, Owen Fahey in the lab developed what is called RefMet, which allows us to harmonize all metabolomics data. Imagine you do a metabolomics experiment, I do a metabolomics experiment. If you want to compare our results, we need to have standardized ways of nomenclaturing, naming the metabolites. We need to have standardized way of analyzing metabolites. And towards this, we created what is called a reference metabolite standard, very much in the same tune as RefGene, which was created by NCBI towards having a common standardized nomenclature for genes. So we created the first nomenclature for metabolites, which is widely used now. It is now adopted by a very large number of national and international resources. It is also accessible through things like PubChem and KEGG and other resources across the country. And so you can, using this, you can search, you can of course review the data, download the data and all that, but you can also do search mass spectrometric data for reference metabolites. You can look at the entire metabolite database and look at the mass spectrometry with exact, search for the mass spec. And we developed this first. In fact, your ability to search the Metabolomics Workbench using mass spectrometric standards is now become very common. So RefMet is a standardized metabolite nomenclature system. If you want to refer to a publication, we published this first in Nature Methods in 2020. It's been now it's considered the standard for nomenclaturing metabolites. RefMet essentially connects to every possible other resource, including INCHI keys, KEGG IDs, PubChem IDs, and all of those. it is a standardized nomenclature. I'll walk you through very quickly what it does. In fact, we have 4 different annotation levels for RefMet. This includes a complete structure level information, which includes regiochemistry, stereochemistry, and bond geometry. It tells you whether it is R or S connected to a given optical isomer, configurational isomer. It tells you about stereochemistry of a double bond, whether it is Z or S, and it also connects you to the entire bond geometry in detail. Then we have some metabolites where you don't have this level of stereochemistry from mass spectrometry data or from other types of measurement data. We use regiochemistry, which provides you with known double bond positions and stereochemistry, but that excludes the bond geometry, doesn't tell you whether it is Z, Zusammen, or opposite, or R or S. And then it gives you the general structural formula. We have the level 3, which is molecular species level, which comes from just simply mass, not MS2. And these are just information on structural features. Complete regiochemistry is unknown, which means you would not know exact location of functional groups or the double bonds. Then you have species level information, which is the lowest level of information, which is just number of metabolite, what metabolites it is, what class it belongs to, etc. So if you look at, for example, if a person doing a study submits metabolite information, this is translated automatically now. We have harmonization systems, which converts, for example, this is the input, which is converted into exact RefMet names, RefMet formula, RefMet, so that we can harmonize with someone else's data that is coming forth. So this is one of the first schemes that enables harmonization of data across multiple studies. So for example, RefMet plays a central role in metabolomics. It connects to all metabolites detected in studies linked to RefMet standard names. It connects to Lipid Maps portal, which I mentioned to you before, through classification. It connects to a metabolite structure database. We have a structure database which provides you concrete ability to look at all structures. All of this is interactive on the website. You don't need to learn any new programming languages or tools or anything like that. And we provide you structured details for every metabolite. It provides links to other ontologies, including KEGID, PubChem ID, etc. It links to pathway databases, including SMPD, HMDB, KEG, Reactome, and many other resources, wiki pathways. And so it provides a complete link to those. And it connects to all collaboration subsets. We have connection to GNPS and other resources, metabolites. And so it connects us to different collaboration sites. So we want to search, if you want to search for unique mass spec in NMDR, which is called, in fact, the Metabolomics Workbench houses National Metabolomics Data Repository, which is the part of the Workbench. The Workbench is more than just a database. It can include tools and user interfaces. We have user interface, for example, to search for a mass spec data or features across a large number of, all the, hundreds of thousands of mass spec studies that we have. And this is using a tool developed in UNC, University of North Carolina, by our collaborator. It's called ADAP-KDB, and this tool allows you to take raw data, all the raw data, convert into open format, detect peaks, does a spectral deconvolution, aligns it, and then does library matching with a NIST. So this is a really useful tool for the community. We have ability to, for example, display in a data site which is called a natural product site, a collaborative site that's run by Peter Dorstein in UC San Diego. It's a large site. We're able to now search for our integrated mass spec data across using GNPS resources. And we can run also these resources through ADAP-KDB that I mentioned to you previously. We can look at, in this collaboration, we can look at, for example, display data in GNPS from Metabolomics Workbench. We can display the mass spec. We can display the sample, and we can look at the dashboard and connect to all the mass spec data in the world. So this is a really unique resource for the community. So next, I want to walk you through the power of analysis tools that we offer in the Metabolomics Workbench. We can start, if you're a newcomer into metabolomics, and if you bring your data, You can do data pre-processing in terms of peak picking and deconvolution and matching and alignment, as I showed you. You can do statistical analysis. You can do chemical property detection, prediction, build a compound libraries based on in silico or in vitro experiments. I should remind you that the Metabolomics Workbench has, over 1 million virtual, compound libraries that is present. And then this can link to metabolite identification. You can identify all the features for targeted, untargeted mass spectrometry, for example. You can do functional analysis. This is one of the unique powers of the Workbench. If you just get a laundry list of metabolites, you can convert them into metabolic information. How does this metabolite, what class it belongs to, what function does it do, how does it connect to pathways, how is it involved in normal physiology and pathophysiology, and then we can do metabolic modeling and in terms of building networks. So the sample, the analysis foundations, this is a paper we published in Analytical Chemistry with collaborators. The sample is really processed. For example, you get LCMS data, MSGCMS data, or LCMS-MS data. We can look at all the features, tens of thousands. We could measure ions, M over Z and retention times. We can do both targeted data processing and untargeted data processing. I'll tell you in a minute more about this. And then you can look at feature detection, tell you, for example, this feature belongs to this mass spec feature belongs to 25, 27 dihydroxy cholesterol or things like that. And then you can look at custom methods for doing annotations of your own. And so this is this toolbox we have developed. In fact, this is a very unique one-stop shop environment. We can go to the Metabolomics Workbench and you can look at, for example, take your metabolites, look at a pie chart of what classes they belong to. For example, you can say these are sphingolipids, these are sterols, these are sterol esters, eicosanoids. You can look at all the classes. You can look at superclasses. You can look at main classes. You can look at subclasses. You can look at averaging, normalization, and averaging. You can look at, for example, all standards. So you can create various types of normalized metrics for analysis. You can do univariate analysis. You can do all kind of plots, perform ANOVA analysis of variance. You can do clustering and correlation, hierarchical clustering, heat map cluster analysis, perform clustered correlation, perform network analysis, do principal component analysis for multivariate, linear discriminant analysis, partial least squares discriminant analysis. We can do OPLS, DA, random forest methods. You can map it to KIC pathways, HMDB pathways, and I'll show you a unique tool we developed called MET-ENP, which provides metabolite enrichment and pathways. And all of this can be done interactively on the Metabolomics Workbench. The data does not have to be already present in the Metabolomics Workbench. You can upload your own data and then integrate it with existing metabolomics data and do the entire analysis on the Workbench. In addition, we provide tools for you to download, and you can do the analysis in your own desktop or a laptop computer, even on an iPhone or an iPad. And in addition, you can also use the cloud environment. We provide a cloud tools. We provide Jupyter notebooks and or Shiny apps for most of these tools. And so you can use it in a cloud environment. For example, you can do the Google Cloud or Amazon Cloud, and you can run these scripts so that you can perform analysis on very large amounts of data. In fact, some of our collaborators with their thousands of studies, they use the Amazon Cloud or the Google Cloud to perform such analysis. Our Stanford collaborators, in fact, they do it very well for the personalized genomic analysis. So I'm going to walk you through two examples in the next 15, 20 minutes to give you an idea of what you can accomplish in the Metabolomics Workbench environment. And it's mainly for people who have not, used the Metabolomics Workbench, and to show you how powerful it is for you to do this. Before I continue on, I don't know whether there are, Kovey, I don't know if there are any questions that audience has. If there are, I am happy to stop and respond to it. We continue to add tools. We continue to add data. But if there are any questions, I'm happy to answer before I go to two really good examples. And if time permits, I'll go to even a third example.
Yeah, there are two questions in the chat. So first question is from Dr. Pendrovsky. So how do your users cite metabolomics data or Metabolomics Workbench?
Thank you very much for that question. I was wrong in not explaining this to you. On the Metabolomics Workbench front page, we provide you with specific instructions on how to cite Metabolomics Workbench reference as well as the source data in terms of the authors who submit the data, their Metabolomics Workbench ID, and also who submitted the data. We tell you precisely how to cite these things. And if you can't find it, please use the help desk to post a question on the help desk. How can I cite this particular data set? And we'll be happy to provide you with this information. And I should add a small anecdote to this. Till very recently, we never insisted on people citing any data, which means that we have not received, in fact, so much Metabolomics Workbench data has been downloaded and used. We never received any credit for having this data. And NIH told us, hey, you need to get credit because we need to have measurable. So NIDDK told me that, hey, please do get people to cite your data. So now we have started suggesting to people that please cite our data.
Okay. Is that answer your question?
Yeah.
Okay. Okay, so Dr. Pendrovsky said can think about using our ID, Research Resource Identifier.
Yeah, thank you. Yeah, that's a very good idea.
Okay, and then there's the second question from Dr. Swampata. Can we link metabolomics data with gene expression in Metabolomics Workbench? That is first based on metabolites abundance data can be determine activity of transcription regulatory pathway. The second one is integrate metabolism and the transcriptome data when both data is available on same samples for mechanistic understanding.
I'm going to provide an answer to both questions in the coming two examples. The two examples I'm going to walk you through shows you how to connect multi-omics data together. And if time permits, I'll even show you a third example, which connects not just simply transcriptomics, but also epigenomic data. So in the next two examples, I promise to answer your question.
Okay. Any other people have any question during this part? You can unmute yourself if you would like. Okay. So Shankar, I don't see other question right now, so you can moving forward. Mm-hmm.
Thank you so much, and thanks for that second question. And this is I'm a systems biologist, so therefore I really care on how we integrate metabolomics data with other types of data. So I'm going to walk you through developments which allow you to do that. This example from human liver cancer, we presented it first in a summer workshop last year. As an example, we developed this for the workshop, and it was a walkthrough. We actually did a hands-on session where people walked through this, and we also gave two other examples, which I will not have time to tell you about on this particular mode. But in the summer workshop video lectures are available on the Metabolomics Workbench, so please feel free to go and look at the summer workshop. So the question that came up was, in fact, this was posed to me by one of my colleagues, and this is what led to this example. What are the mechanisms and drivers of hepatocarcinoma in humans? And this particular individual was interested in gene lists in hepatocellular carcinoma. And he wanted us to really take gene lists and then try to figure out how is metabolism coupled to this gene list, and what can we learn about metabolic pathways, and what can we learn about going beyond the transcriptome and the metabolome. So I'm going to answer all those questions. First, I want to get you to come from gene lists into metabolomics. We have a metabogene. We have a complete list of mammalian genes which connect to metabolism. In fact, there are about 2,000 between 1,000 and 2,000 human genes which are involved in myriad metabolic pathways. And we have all of those listed as a database which can be accessed. But in any case, in addition to that, we developed something called MedGene. This directly answers your question. This is a connection between gene-centric metabolomics information retrieval tool. That's what MedGene stands for. It's available both online. It's available for you to download as a source code and run on your own computer, both in R and Python. mode. We also have a cloud, a Shiny app, which allows you to run it on the cloud. So MedGene allows you to connect, as I'll show you in just a minute, connect metabolites and genes. Then we also can connect to the Common Fund Data Ecosystem Workbench, which I'm one of the PIs on. And I can show you how we connect to discrete data like glycomic data, drug data, and other types of data. So we really provide a complete pipeline for you to connect all the way from the genes to metabolites or metabolites to genes. And so you can identify all the metabolomics workbench studies, for example, connected to hepatocarcinoma, connecting these genes, the gene list I'll show you in a minute, and apply various analysis tools, including statistical tools and metabolite enrichment and of pathways. And then you can visualize, interpret the results, generate hypotheses, and then download all of these. And then this is really the whole power Metabolomics Workbench offers. So I'm going to walk you through this example. This scientist came to us and said, I got a list of genes that I'm interested in, which are implicated in hepatocellular carcinoma. There's first gene is PKM, or PKLR, pyruvate kinase. It's significantly upregulated in hepatocellular carcinoma. It's involved in tumor growth and is associated with poor prognosis. And it's involved in germline loss of PKM2 promotes metabolic distress and hepatocellular carcinoma. Effects of PKM2 on global metabolic changes and from gene expression to drug discovery. These are known. These are links to papers which are recently published on these. The second gene this person was interested in was MAT2A, methionine adenosyl transferase, involved in regulation of S-adenosyl methionine, and it results in degranulation of methionine metabolism, and it's a marker of progression and prognosis of hepatocellular carcinoma. And this recent papers in this include deregulation of methionine metabolism and translocation of this relevance for hepatocellular carcinoma. The third gene this colleague was interested in was acyl-CoA synthetase long chain family member 3, which along with MAT2A forms a subpathway that protects cells from ferroptosis, a type of cell death. And this pathway is particularly active in hepatocellular carcinoma, helping cancer cells survive under metabolic stress. And these are three genes which are really involved in hepatocellular carcinoma. And the question came up, how do they alter metabolism? And is there a way for us to really build a systems level story of what happens, what hypotheses can be formed, what new experiments can be done? So I'm going to walk you through this. So we said, okay, here's MedGene. So you can go to this website where, in fact, it's just called MedGene. You don't need to do all this. This is specific to this particular question we posed. In MedGene, we asked the question, 3 genes we gave simultaneously. You can give a whole list of genes. You can give 1 gene or 10 genes or 100 genes here in gene ID package. So we gave MAT2A, ACSL3, and PKLR, PKM. And we gave this gene ID, and we said, okay, let's find out what metabolites are involved. MedGene gives you connection to human. You can connect to any species, human, etc. We chose human, liver, cancer. These are all annotations of these. You can look at pathways. You can look at reactions. You can look at metabolites. You can look at, and I'm going to walk you through these. So when we try to identify all the metabolites that are involved and on the metabolomic studies available in the Metabolomics Workbench, connecting these genes, you can see immediately there are many studies in the Metabolomics Workbench which couple to these, and these are the metabolites largely involved. I mean, phosphoric acid, S-adenosyl methionine, coenzyme A, AMP, pyruvic acid, etc. So these are all the different metabolites that we can couple to these genes. Then when you look at the studies, let's take an example study. For example, there are 2 studies. One is a study involving hepatocellular carcinoma, comprehensive analysis of transcriptome and metabolome in intrahepatic cholangiocarcinoma and hepatocellular carcinoma. And then this is a part A study and part B of the study, 230 and 231. If you take these 2 studies as exemplifying the genes that are present and the metabolites associated with those, we can go into look at all the metabolomics data that's available currently for these different genes. You can look at the data set, for example, ESI electrospray ionization negative ion mode. You can look at a pie chart. You can look at all these different types of analyses, and I'm going to show you a few of these. You can do a pie chart of metabolite main classes present in this study in cancer in comparison to control. And you can look at the study, and immediately you find the studies that the largest number of metabolite classes involved are amino acids and peptides. And then there is purines, there is fatty acids, there's all kinds of other species. And these are all the numbers of the species present in the hepatocellular carcinoma in contrast to controls. And so if you can look at the same thing, imagine I want to compare the tissue type of hepatocellular carcinoma versus non-tumor surrounding tissue of HCC. If I want to compare these two groups, I can do a comparison with volcano plots. I can look at a metabolite class level volcano plots and identification. For example, I can look at all the significantly enriched metabolites, positively enriched and negatively enriched or negatively regulated. I can look at a comparison between these. I can download this data, and then I can further look at it in the form of volcano plot analysis further in terms of saying, hey, these are all the amino acids. I can download a list of these. These are all the nicotinic acid alkaloids, monosaccharide phosphates, dipeptides that are upregulated. I can look at all kinds of statistics, including class enrichment by Kolmogorov-Smirnov test. I can do analysis of variance across if you have multiple studies to look at whether this is significant. I can do a whole series of analysis. I can look at heat maps of this, and then I can look at a contrast between this ESI for ESI negative ion mode and positive ion mode. I can do a comparison, a heat map of all the metabolites that are present, clustered them, and then you can see the enrichment of these classes of metabolites, these metabolites in the negative mode and the positive mode. And this is actually a publication quality figure. You can download it in terms of high resolution figure to show how the comprehensive analysis of transcriptome and metabolome in intrahepatic cholangiocarcinoma and hepatocellular carcinoma emerge as being correlated. We can look at OPLS-DA analysis. I'd mentioned to you that we can look at least square analysis. And then you can see clearly there are two groups, group 1 and group 2. Group 1 is the carcinoma group, cancer group. Group 2 is the non-cancer group. And we can look at all the scores for these metabolites that are associated in this group 1 and group 2. You can do further analysis of these in the data. And you can do, for example, you can download all these data files. You can download the process files. And it essentially provides you with a very rich environment for you to do a comprehensive analysis. So we applied MET-TNP to another data set, the same data set. And when we look at comparison here, for example, we provide you also what does metabolite enrichment and pathways do in terms of analysis. And we provide you a tutorial for looking at all this data. You can look at this data and look at the MET-TNP results, enrichment results. You can show, for example, what functional pathways are enriched. For example, here you have monosaccharide phosphates pathways involved, for instance, and you can then convert that into pathway diagrams, connection diagrams, by looking at complex connection of what metabolism is altered in this hepatocellular carcinoma. We can look at the volcano plot if you want to look at what are the enriched metabolites. You can convert that into a metabolomics map of enriched pathways. So you can already see the power of doing this types of analysis with the Metabolomics Workbench. And in this tutorial, we also went to, how to connect this, for example, to other data. I promised you that we would not only connect to genes and metabolites or transcripts and metabolites, but we also connect to glycoproteins, etc. So we created what's called the Common Fund Data Ecosystem Workbench. And the workbench, you can enter this name S-adenosyl methionine, which is one of the metabolites involved. And it tells you, for example, glygen, which is a glycomics resource, has information on the glycosylation of proteins involved with S-adenosyl methionine. And you can go to glygen, and we allow you to download the data from the glygen portal if it is public. And so you can then analyze it further. We can go and look at, for example, obtain data from other Common Fund projects. For example, you can get information from glygen, from extracellular RNA. You can look at what extracellular RNA is involved in, along with these genes and metabolites in hepatocellular carcinoma. So you can provide a list of those. So this really provides you with a dramatically rich environment for looking at results. And you can narrow down. In fact, we provide you filters for you to look at only certain comorbidity diseases, etc. For example, hepatocellular carcinoma and diabetes, or any other type of disease. It provides you data from multiple resources for you to visualize, download, and analyze. So it's a comprehensive environment for analysis. So we can view the project information in the portal. You can download it from the portal. For example, you can go to the mass spectrometry data, and you can download the mass spec data that I mentioned to you previously. So we can view the information directly on the Metabolomics Workbench or on the CFDE Workbench. We provide you with a mutual connection between the two. And we provide you with, for example, the gene expression data. Somebody asked me the question, can you get gene expression data? Yes, we can. In fact, for the gene that you mentioned, I mentioned acyl-CoA synthetase long-chain fatty acid member, we can go and get the gene information, gene expression information from links or from any other GTEx, other resources that are present. Same thing for other genes. So that's the first example I wanted to show you how you as an end user, without even involving us, can really go in and try to do a whole sequence of analysis using the Metabolomics Workbench to come up with new hypotheses for you to test. And before I go to the second example, which should be very quick, I want to pause and ask questions if you have any questions for me.
Yes, there are questions from the chat. So the first question from the audience is how does MedGene take into account issue with problematic cell lines, including HepG2, which was first identified as a hepatocellular carcinoma, but is now known to be a hepatoblastoma? The cell line was used in 22,000 papers.
Yeah, thank you very much. I mean, disambiguation of biological, hypothesis and premise is always a very, very complex issue. We in Metabolomics Workbench do not make judgments. We don't tell you whether this is a faulty, this particular use of HepG2 is faulty or so. However, in the CFDE Workbench that I showed you, we provide you annotations. We connect you to also pertinent literature, which allows you to make your own judgment in terms of saying, should you use a cell line or not? Having said that, I just want to point out, I'm not a great aficionado of cell lines, of derived cell lines or immortalized cell lines, because they often lose their original perspective, and they are so transformed into a different cell line that they lose their native phenotypic abilities. And so be careful when you use immortalized cell lines. This is what I have to say in general to biologists. But we in Metabolomics Workbench do not make any prescription on what you should use and should not use, because that goes beyond the remit of what we are supposed to do. As a scientist, I will do it. If you call me and ask me, I'll tell you, hey, should you do it or not? But in the Metabolomics Workbench, we don't provide this curation capability.
Does that answer your question, or?
Yeah, I'm just wondering what the data is based on in Metabolomics.
The data I showed you is the data based on submission to Metabolomics Workbench and on gene expression data submitted to different resources like links, GTEx, and others. And so we integrate in the Common Fund data ecosystem, we integrate all this data. In MetENP, we connect in MedGene, we connect NCBI data and metabolomics data. In MetENP, we connect gene expression data where available. And in CFDE, we connect gene expression data for you to look at also other types of data like glycomic data, drug data, and so forth. I hope that answers your question.
Thank you.
Okay, so move to the next question. It's from Dr. Swambada. How the tissue cell type specificity of metabolic reaction, metabolism, regulatory gene abundance, and kinetics are modeled in the Metabolomics Workbench database?
Yeah, thank you very much for that question. So we annotate all our data as a source of the data, as being either whole liver or cells, primary cells, or immortalized cells. So we have annotations of those. So if you choose a data set from a cell line, then that will depict what is available in this. The reactions will be depicted as what belongs to that, pertinent to that data in that cell line. If you choose, for example, if you choose data from the whole liver, like I'll show you in the next example, then it pertains to reactions in the liver. It pertains to data in the liver. And so depending upon the choice of data sets, we will allow you to, the MetaENP allows you to do the analysis with reference to that data. If you ask me a more complex question, can I integrate a cell line analysis with the primary cell or with the tissue-based analysis? The ball is in your court because you're the scientist who decides what you want to integrate. If you think the integration makes biological sense, then we let you do the integration. We don't prescribe to you whether the integration makes sense or not. Whereas if you ask me as a scientist again, I'll tell you whether it makes sense or not. But as a data resource, as an integrative resource, we don't make that judgment for you.
Hope that answers your question, Dr. Da. If you would like to discussion, you can just unmute. Yeah. And then, okay, that's great. So the next question is from Dr. Krishnan. Is it also possible to link non-coding RNAs, such as microRNAs, to metabolomics data using the Metabolomics Workbench?
We are just in the process of beginning to establish such a link through the Common Fund data ecosystem. So we are trying to integrate all the data available. So we're just, it's in the early stages of us to think about microRNA tool integration. And so, for example, we want to look at MIR-base or one of the MIR resources to integrate that. We don't have it yet available at this point in time.
Okay, and the next question is from Dr. Song. Does Metabolomics Workbench provides any untargeted metabolomics data, such as the chemical features, not just identified features?
Oh, absolutely. In fact, we have we have tons of untargeted metabolomics data. And we have tools which allow you to really look at where features are available. And we also provide you where retention time is available and M over Z values are available. We help you identify those things. So no question. And our collaboration with GNPS, we just published a paper in one of the Nature Family journals on showing how our resources collectively, this is Peter Dorstein, is the senior author, a primary author on that. He shows how you can really integrate across different data types towards analyzing untargeted mass spec data.
Okay, and then he has a second question: were the quantified result normalized by any method, internal standard, TIC, etc.?
Excellent question. So we provide where people have used standards and provide quantification, we allow you to do the quantification. We have multiple normalization strategies. If you go to the toolbox I showed you, there are multiple normalization strategies for the data. And we allow you to choose your own normalization strategy. And we have in our own curation, we have a default normalization approaches we use, but you are welcome to choose a different approach on the workbench.
Okay. Yeah, so that answers his question. I don't see any other question right now, so we can move to the next session.
Thank you. So let me take the last few minutes. I thought I'm going to go walk you through two, really complex examples, but I won't have time. I'll walk you through one of those. So this is work, in fact, Dr. Ed Dennis, who is in the audience, will appreciate this. This is part of the Lipid Maps initiative. We looked at fatty liver disease. And this also shows the connection between genes and lipids that you asked me about. And this specific one was a collaboration with our colleague, Alex Brown, the late Alex Brown. Unfortunately, he passed away, amazing scientist, and it's really a sad tragedy that he died young. But it was a collaboration with him and with Gordon, who was involved in terms of doing the initial sample clinical study that was associated. The clinical study involved 4 different, the tissue samples from normal liver patients, from simple steatosis and NASH patients, and the NASH patients, non-alcoholic steatohepatitis patients, and cirrhosis patients. So we had this normal steatosis, NASH, cirrhosis patient recruitment in Vanderbilt. This is all done in Vanderbilt. They took 31 normal patients, 17 steatosis patients, 20 NASH patients and 20 cirrhosis patients. And we looked at, in fact, the measurements were done across lipid maps, across multiple investigators and lipid maps for all the lipid classes and lipid studies in the liver tissue and in, I believe, blood tissue, and then plasma. And then when you compare the common metabolites between liver and plasma, which is one of the goals of this, was to ask the question, hey, can we really get information from plasma about different diseases? And can we discriminate, for example, between fatty liver disease and steatohepatitis? If you can do that from blood in correlation with what happens in the liver, it'll be fantastic. So we identified, in fact, 48 common species between liver and plasma. I'm highlighting most of them here. And these species were really highly correlated between plasma and liver, and the liver across these different 4 conditions that I highlighted. And so if you look at the heat map of these different conditions, for example, steatosis compared to normal, NASH compared to normal, cirrhosis compared to normal, NASH compared to steatosis, you can already see differential regulation of different metabolites represented in the heat map format. Of course, we can do statistics and quantify these things. And if you do statistics, you can really look at discrimination using a discriminant analysis. You can look at, for example, you can look at blue is steatosis. Steatosis separates out from black, which is normal, which separates out from orange, which is the steatohepatitis, which is here, and then the red is cirrhosis. So there's a really good separation using these different metabolites that are common between the plasma and the tissue. And in fact, when we look at the discriminant score, we can see a really beautiful separation and a linear discriminant score if you take these metabolites that were discriminated, these key metabolites, something like 18 or so, phosphocholine, phosphoinositols, phosphethanolamines, and ceramides. And you can see clearly that there's a good discrimination between those. And this is really very useful. Some of my clinical colleagues were really excited about this fact that 20 plasma metabolites were essentially able to discriminate between fatty liver disease and steatohepatitis. And if you look at liver, somebody asked me the question about liver transcripts. How are these correlated with transcripts? If you look at liver transcripts, you can see clearly, for example, the genes which are involved, for example, in formation of fibrotic tissue, this going up between normal to steatosis to steatohepatitis to carcinoma. You can see a significant progression in the collagens, which are representative of more fibrous tissue. And then if you look at all the genes that I highlighted, metabolites I highlighted to you, and the genes involved in inflammation, you can again see a significant increase in progression across the disease. For example, liver transcripts show upregulation of disease progression. MMP14, for example, shows between cirrhosis and other disease stages. Look at MMP14. This is all, going towards the cancer. So we are really able to use integration between transcriptomics and lipidomics, but we can also integrate genomics. In fact, look at mutations. In fact, we look systematically at all the mutations, the single nucleotide mutations. And if you look at that, you can see that many genes are showing both synonymous missense and then other missense mutations. But most interestingly, we identified this gene that was made famous by Helen Hobbs in Texas, that she identified PNPLA3 gene, PNPLA3 to be a gene significantly involved in differentially regulating steatohepatitis in European population. And so we were able to identify this gene and its implications in metabolism. So I just wanted to give you an idea of how you can integrate these multiple different approaches, different data sets towards producing significant biological insights, mechanisms, and new hypotheses. Unfortunately, I've run out of time. Otherwise, I was going to tell you a story between atherosclerosis, connection between metabolomics, transcriptomics, and epigenomics, but I've run out of time. And maybe if Kovei invites me another time, I'll be happy to tell you in more detail about our connection between these different omics in atherosclerotic disease formation. And so we did extensive amount of work in this area, both transcriptomics and metabolomics. We published a large number of papers, in collaboration with investigators, both in UCSD and outside. And this provides us, again, with an ability to bring metabolomics into the central focus of regulation or in pathology. And so I hope I've convinced you that Metabolomics Workbench does offer a unique opportunity for you to have an integrative platform, along with other resources like Common Fund Data Ecosystem Workbench, to provide really deep insight into human physiology and pathophysiology. I'm happy to stop and take any questions at this point in time. I won't go into details of these. This is really work that you'll appreciate. This is all the dramatic number of experiments we did. And I will not have time to tell you about these. And so I will pause at this point in time and take questions from you folks. And hopefully you will enjoy the Metabolomics Workbench.
Yeah, thank you, Dr. Subramaniam. So there is a question from the audience about that section. So it's from Dr. Edward Dennis. So you have emphasized where gene upregulation define disease and then looked for metabolic change. Are there good examples where metabolite change in disease predicted gene upregulating and was then verified?
Yeah, I mean, in literature, Ed, thank you for the question. In literature, there are many, many examples where metabolic changes have given us a gene upregulation. And a very good example is in our own study in lipid maps. If you recall the statin work, we showed that interference with statin involves the regulation of LXR and RXR and other ligand receptors. And also it involved prostaglandin E to increase in NF-kappa B and prostaglandin expression. And there's a strong metabolic connection. I also want to point out to you that, if you look at epigenetics and epigenomics, look at histone modification, acetylation and methylation of histones are often regulated, connected, very strongly coupled to metabolism. And again, another study from Chris Glass's group jointly with us, and if you recall this, is the desmosterol, how it regulates the regulation of genes. And the story I was going to tell you about atherosclerosis, I actually drew a direct comparison between metabolism, cholesterol metabolism, and regulation of KLF4 gene in endothelial cells and in macrophages, and LXR genes, and also cholesterol 25 hydroxylase genes, how they were regulated. So that was published, in fact, jointly in collaboration with John Shee's group recently. And we showed, in fact, a strong coupling between metabolism, epigenetics, and transcriptomics.
Does that answer your question? I think I see Dr. Dennis, you're unmuted.
Ed, you're muted, so you can tell us if it makes sense.
Yes, thank you.
Okay, thank you. And then we have next question from Dr. Da. Just to thank you to let you know we use Metabolomics Workbench as well as a GSEA developed by you.
Thank you.
And then the next question is from Dr. Gonzalez. Can I upload my quad table from GNPS and perform the downstream analysis directly on your platform?
Absolutely. Absolutely. We already have the tools and ability to do that. Absolutely.
Okay. Any other people have question? You can just unmute yourself or raise your hand. Join this discussion.
I see some questions, and we do provide untargeted metabolomics data. A large amount of data we have is untargeted metabolomics data.
Yeah.
And I want to explain to you something very important before I let you folks go. We actually provide metadata. I mean, we demand that users who submit data submit metadata, because in my opinion, without metadata, the data does not make sense. And so therefore, we provide metadata. You can use the Common Fund Data Ecosystem to filter your studies based on all the metadata we provide. We provide you with a very beautiful interface for you to do that. And if you're not seeing it, take a look at it. So you can filter your data and pick only the data that's relevant to you. And finally, I should say we got an award from FAIR, which is a findable, accessible, reusable, interoperable, and reusable resource. FAIR said that we are one of the most fair resources available in the community.